Hello, fellow foodies, and welcome back. This is Dr. Cassandra Quave, and you're listening to Foodie Pharmacology, the science podcast for the food curious. This week on the show, I'm really excited to have a very special guest who's going to explain to us a bit about how and why plants make medicines. And we're talking about medicines that humans can use, and really what's the machinery behind that production pathway. So, our guest is Dr. Ryan Nett. He is an assistant professor in the Department of Molecular and Cellular Biology at Harvard College. He grew up in rural Illinois and earned his bachelor's in biology and chemistry, and then went on to complete his PhD in the lab of Dr. Reuben Peters at Iowa State University, where he investigated the biosynthesis and biological function of plant hormones produced by plant associated bacteria. Then, as a postdoc with Dr. Elizabeth Sadley at Stanford, he studied the biosynthesis and engineering of medicinal plant alkaloids. Today, he runs a lab focused on understanding the unique chemistry and functions of plant molecules. His work has been recognized by a Smith Family Award for Excellence in Biomedical Research. And most excitingly, his latest news is that he was just named as a Pew Biomedical Scholar. When he's not in the lab, Ryan enjoys playing basketball and foraging for edible mushrooms with his wife and their three children. So, welcome on the show, Ryan. It's great to see you.
Hey, Cassie, thanks so much for inviting me.
Yeah. I mean, there's so many questions I know we could dive into, and maybe we can start with the basics. Maybe what is plant biosynthesis and why do you study it? Let's start there.
Yeah, so when we're talking about biosynthesis, we're referring to the natural mechanisms that plants use to build molecules. And this could really be anything, any molecule that's important for the plants or that is important to us, whether it's a flavor or a fragrance from the plant, or a lot of the things we focus on in my lab are small molecules that have medicinal properties. And so really what we're trying to understand are the chemical pathways that plants are using to make the compounds that are useful. And so this is both finding proteins, enzymes that act like molecular machines to catalyze chemical reactions, and then actually be able to put those reactions together so we can go from some building block, some metabolite that we know a lot about into a more specialized molecule that has some useful property. And one of the motivating factors for understanding this, other than the fact that plants have really amazing chemistry, and I think we'll talk about that in more detail later on, is that there's often a need to understand these biosynthetic pathways because molecules can be hard to source from plants for a bunch of different reasons. Plants can be hard to access, slow growing, endangered. They might produce the molecules at low levels. And we feel that if we can understand how plants make the molecule, we can take those principles and then engineer the production of the molecules in other systems that might be more tractable or cheaper to produce a molecule.
It's incredible. And so, maybe just to give some folks some background, I'll talk about one of my favorite plant molecules, caffeine. It's an alkaloid produced in coffee and tea. So, these are molecules that plants produce kind of to defend themselves, right, from insects or herbivores. Um, why else might plants make these very specialized molecules?
Yeah, I think on the surface, it seems so many molecules, caffeine's one, for example, that has actually been studied and has been shown to be directly related to plant defense. But I would say for really the vast majority of these molecules that we think about as like natural products, like small drug-like molecules, we don't necessarily have a good handle typically on what they're truly doing for the plant. We can speculate often, like if something is toxic to us, humans, there's a good chance it's toxic to other mammals. Also, there's a chance it's toxic to insects or things that share similar biology to us, whether they're animals or otherwise. But defense is a very common one. I think a lot of the molecules we work on in my lab are alkaloids. These have historically been looked at as defense-related molecules, in part because many of them affect neural cognition in humans, and it's pretty easy to sort of relate that to how that might convince an animal not to eat a plant anymore if it has a really bad experience. Um, in many cases, these alkaloids are just very toxic. There's famous examples. Socrates is poisoned by a plant alkaloid, and so it's kind of we can easily make sort of the connections that these are probably involved in defense. But even something like caffeine, I think, depending on context, can have different functions. Obviously, it's not defensive against us. We sort of enjoy its properties. But even for insects, where it can be defensive in, say, the leaves of the tea plant, it's also a molecule that's found in other contexts, like in the nectar of citrus, where it can actually be something that might manipulate the behavior of bees to make them come back to that plant and pollinate it more. This is all dependent on the concentration of the molecule and where it's located and things like that. But even beyond these interactions with insects, I mean, plants are in competition with all organisms, including other plants. So there's lots of examples of plants secreting molecules into the soil that will inhibit the germination or the growth of other plants, a term called allelopathy. So this is something that's quite prevalent in the plant kingdom. You can imagine fighting for soil space as well as for access to sunlight is quite important.
That's wild. Just
Yeah.
for a second, for folks to reflect on that. If you have let's say there's a tree and this tree wants to defend its territory, its resources, it basically releases molecules into the soil that prevent other species from germinating. So it like kind of stakes its claim. I think that's something a lot of folks don't quite appreciate is just how communicative like plants are. They're constantly producing these molecules to signal with not only other plants, but also other organisms across different kingdoms with animals, with insects. Yeah, keep going. This is fascinating.
Yeah, of course. I mean, I think what people often point to with plants in that regard is they can't move. So like we think of animals and the ways in which they can adapt to challenges in their ecosystem, their environment. One common strategy is to go hide or run away or even like physically defend yourself. And in the case of plants, for the most part, they cannot move. They're kind of stuck in place. And you think of something like a tree, it needs to be in that place for decades, or in some cases, hundreds or thousands of years, depending on the tree. And so they need ways to interact with their environment, whether they're defending themselves or communicating with something that's beneficial. So whether it's defense, secreting things to the soil to inhibit other plants from growing, this can also be recruiting beneficial microbes in the soil, bacteria and fungi that can form useful interactions with the plant for providing nutrients. I mean, you mentioned up front, I studied nitrogen fixing bacteria when I was a graduate student. These are bacteria that form very close association. Plants provide them nitrogen. And the way that they form those interactions is all through chemistry, molecules that the plant is secreting to the bacteria and vice versa in order to communicate. And then, of course, there's examples everybody's familiar with, like pollinators and the nice scents that come from flowers and fruit to attract insects and animals to come either pollinate or disperse seeds. And so really being fixed in place has led plants to develop all of this chemistry that they can use to communicate, often in ways we can't see, but it can be very impactful, obviously for the plant, but also for all the organisms that interact with plants, including us.
Incredible. So I want to talk a little bit about plant genetics and to give folks a bit of background. So when it comes to mining natural products from microorganisms, a lot of scientists study something called biosynthetic gene clusters. And that's because in certain microbes, you can have clusters of genes located physically close to one another, and you can isolate those clusters and really study and kind of um, work on expression of those genes to yield a higher amount or manipulate the production of your target molecules. But with plants, it's a bit different, right? What can you tell us about plant genetics? Do they cluster? And if not, what are the challenges that that presents?
Yeah, so there's a couple of challenges here. One is, or historically, and I think still to some extent, has been just getting the genetic information. So for bacteria, microbes in general, their genomes tend to be relatively small. And with current sequencing technologies, this is information you can very quickly put their genomes together. And so actually looking for the genes is an endeavor that can be done relatively quickly. In the case of plants, their genomes can be enormous. In terms of comparing it to the human genome, they can be twice as large, oftentimes much larger than this. They often have polyploidy, so many copies of the same chromosome that complicates things. There's all these repetitive elements that just make it a nightmare to actually get genomic information. And so historically, we didn't have access to a lot of plant genetic material to actually investigate. So that's the first challenge. The second one is you kind of allude to as well. In microbes, you have these nice, tidy gene clusters where you're in all of the genes that encode for the enzymes to build your molecule are right next to each other in the genome. So if you know one gene, you look to the left and you look to the right on the genome, and there's the rest of them. And so in terms of finding the gene, pretty straightforward. For the most part in plants, you don't see this. There are some exceptions. Gene clustering is something that's emerging as more common than perhaps we once thought in plants. And I would say it's probably the exception to the rule. The rule tends to be that these genes are spread out across these massive genomes. And so then if you actually want to find the genes involved in a pathway in plants, not only do you need to get genetic information, which can be hard to come by, but then once you have it, you can't just look for a single locus on the genome and hope to find all your genes. It becomes this sort of needle in a haystack problem. And there's many ways that we can sort of get around this, but it's, I think, led to a major lag in terms of our ability to decipher the chemical pathways in plants compared to microbes, where things have been easier in many regards.
Oh, that's great. I'm wondering if it might be helpful to kind of go over a specific example of a molecule that you work on in the lab. I know you're doing some really interesting work on huperazine A. Can you use that as kind of a case study to walk us through what the process is like when you're trying to study production of a certain molecule by a plant?
Yeah. So I think Cooper's DNA is a nice example, in part because I've spent a lot of time on this going back to my postdoc, and now it's been a focus of my lab. So this molecule, Cooper's DNA, Coupe, I might call it for short, in case you're wondering what I'm talking about. This is a molecule from club mosses. So one of the famous species is Cuperzia serrata. This is a traditional Chinese medicine, but used to treat many things, including different cognitive diseases as well as some forms of pain. But this molecule itself, huperzine, has generated interest because it's an acetylcholinesterase inhibitor. So it acts on an enzyme in the neural synapse of mammalian brains. And this is a major mode of action for drugs that are used to treat Alzheimer's. So we wanted to understand how the plants make this molecule. And really the first thing that we want to understand is where and when it's actually made in the plant. And this is a relatively simple concept, but it's actually quite important for us because these plants are long lived. Club mosses are small, but they can live for decades. And they live in places like Massachusetts that have four seasons. And so they might not be metabolically active all year round, but you could find the molecule in the plant all year round. So probably making the molecule and then storing it. And what we want to do ultimately is try to figure out the genes that are making it. And those genes are probably only going to be expressed where and when the molecule is actually being made. And so we spend a lot of time as a lab upfront in these projects trying to figure this out. Can we profile the plant, figure out where and when the chemical is actively being made? And there's many different ways to do this. People that listen to the show might be familiar with like isotope tracing studies where you put heavy atoms into molecules, you feed them to your organism, and then you see where those heavy atoms end up. That can give you a sense of where active metabolism is occurring. You can also just profile the abundance of the molecule across different tissues. In my lab, we use something called liquid chromatography mass spectrometry, or LCMS. I know mass spec is something that's been talked about many times on the show as well. But in a sense, what we want to do is just figure out where and when is the plant making the molecule. And once we know that, we can then look specifically in that tissue or at that specific time for genes that have enriched expression. And those are the genes that we begin to look at and trying to figure out what's involved in actually, what enzymes are actually involved in synthesizing our molecule.
That's cool. So you're able to use these kind of chemical sensors in the lab to kind of look at different tissues to determine where the molecule's located. One thing that's fascinated me though too, Ryan, is the fact that where a molecule ends up doesn't necessarily mean that's the tissue where it originates. So can you tell us a bit more about like how molecules are transported? Like, is it possible for something to actually be produced in the roots and then moved into the leaves or into the fruit or how does all of that work in plants?
Yeah, so this is something we always worry about in our work, that we could be tricked by just looking at where the molecule accumulates. A famous example of this that's very well studied is nicotine in tobacco, where the molecule is actually synthesized in the roots and then is transported up to the leaves where it actually then accumulates and have this deposited nicotine that people associate with cigarettes and smoking and everything like that. But it's actually made in the roots before it goes there. So when we do these isotope tracer studies, we're really trying to tease apart aspects like that. Like, can we be confident that where the molecule is accumulating is acting actually where it's being made? And I would say this idea of like, I refer to it often as like the organization of metabolism, thinking about not just the chemical reactions, but where they're actually occurring. I think this is something that people have thought about for a long time and made lots of progress on, but it's a very challenging thing to study because oftentimes we can observe that the molecule is being made in one place chemically and then being transported somewhere else. We don't actually know the mechanisms by which that happens. What are the transporters that are involved in moving it out of the roots? What are the transporters involved in actually taking those back in to cells in the leaves. And there's increasing evidence for many biosynthetic pathways now that there are specialized cells littered throughout the plant that might be dedicated to making a molecule. And so if you think of a biosynthetic pathway, maybe if there's 20 enzymes catalyzed in 20 steps, these could all be occurring in one cell type. You can think of it as like one test tube holding all of your catalysts, or they could be separated amongst many different cells. You might have 5 genes in one set of cells and 5 genes in another, and so on and so forth. And this requires movement of metabolic intermediates among all of these cells. This is something that I think is a major unknown for most of the pathways that we work on, and something that can certainly confound our ability to actually like go in and break down what we think is happening biosynthetic.
Yeah. Well, I think it's really I think it's really interesting to see. I mean, if one looks at the physical structure of a lot of these advanced secondary metabolites used in plant defense, but also which have medicinal properties, these can be very strange molecules that are kind of twisted, that have unique chirality or unique functional groups. And I think this is something also a lot of us overlook, is that it's not just one enzyme spitting out this one molecule. There are different enzymes along the path. And for those of you that aren't familiar with what I mean by enzyme, that's like a kind of specialized protein that can modify the structure and add things or pull things off. And they're basically tinkering with the molecular structure along this pathway. And then I think what else is interesting is at the same time, we have environmental cues that the plant is then interpreting, let's say from something munching on the leaves, maybe they ramp up production of some of these enzymes responsible for then scale up of these molecules. There's so much dynamic activity happening at the plant level. And it's really not a simple thing to tease apart. And this is why I'm so fascinated by the work that you do, Ryan, because there aren't that many labs that are really have the skill set to dive into these big puzzles and where these molecules come from.
Yeah, I think you sort of hit the nail on the head with a lot of this. There's a lot to balance all at once. I think we're trying as a lab to be better plant biologists. I think historically there's been a lot of work by natural products chemists and biochemists to understand these enzymes that catalyze reactions for these molecules. It's becoming increasingly more clear that we need to understand more about the actual biology of the plant to put all of this into context. Like you mentioned, a molecule isn't going to be made under all conditions. It might only be induced when you have a microbe infecting the plant or a caterpillar chewing out a leaf or something like that. And so if you go into a plant not knowing that, you might be looking for genes that aren't even being expressed at that time and place. And so, it's unique for each project that we have in the lab, but we really try to understand upfront what are the conditions that are controlling whether or not a molecule is actively being made. Actually, for many of the molecules that we work on in my group, they tend to be highly expressed. The genes tend to be highly expressed, and the molecule tends to be actively being made in very new parts of the plant. So, like new growth, places that might be extra susceptible to pathogen attack or pathogen infection or herbivores. I like to think of when, if you plant a tree, at least growing up, we would travel up to Minnesota for family vacations. We would try to plant trees on the property, and you'd always have to worry about deer coming in and biting off the new buds of the plant because they're probably the most tasty, easy to digest parts. And what we find for a lot of these alkaloids that we work on is that's exactly where they're being made and heavily enriched. Kind of makes sense, at least intuitively to us, that like this is probably a part of the plant you really want to protect. And we've been able to leverage that information to dive in and look for the relevant genes and enzymes that are making the molecules that accumulate.
That's incredible. I mean, this also, in a way, translates so clearly for me to traditional knowledge because, as you know, I've worked with healers all around the world, and they have very specific criteria when they're looking to collect a wild plant, like a wild medicinal plant. They look at the age, or perhaps if it's a liana, how broad is the vine, or what species is it growing next to. There are all these kind of environmental cues they're looking at, and at the surface, you're thinking, well, is this just an old wives' tale? Like, why does it matter what these other environmental cues are doing? But actually, it matters a lot because over time, they've realized that those environmental cues have helped to stimulate production of some of these bioactive molecules. And in a way, I mean, I think we all encounter plant chemistry. I'm thinking in particular of How do we know when a fruit is mature? We have a change in color. Well, color is a change in chemistry. We have a change in flavor. They move more sugars into the fruit because the seed are ready to be dispersed. So we read these signals all the time without really thinking about it.
Yeah, for sure. And I think almost all of our projects, or I would venture to say all of them, probably have started with traditional knowledge in some way, shape, or form. I mean, I can't think of a situation in my lab where we're starting from scratch on something. I mentioned like natural products chemists. This is work that goes back several decades, where at a chemical level, people are trying to understand the specific molecules produced in plants. But the reason that those natural products chemists were typically working on those plants in the first place was because there was some traditional knowledge of some utility of the plant, whether it was medicinal or otherwise. And I think oftentimes some of this can come across as anecdotal, but that's often very useful information. Like we're working on plants called lobelias, which are found all over the world. You can find them along the Charles River here. But if you just go try to buy them from a greenhouse or something, they'll be advertised as useful to plant amongst your garden because they deter rabbits and deer. And this is probably something that's been observed for a long time. And now at a chemical level, we can look at that and be like, oh, well, it's because they're full of these neurotoxic alkaloids. And so it all sort of stems back to just the human observation, things that are maybe very intuitive to people, whether they're scientists or chemists or not. And then we've been sort of able to build on that at a more biochemical.
That's incredible. I'm thinking, for all the gardeners out there listening, this is when we talk about companion planting. It's about selecting those species to help defend your heirloom crops and things like that. Incredible. Well, I want to talk about another example. I know you've also done some work on a molecule called colchicine. Now, this is, if I remember correctly, from the autumn crocus plants that you find this on. A beautiful little plant, but also quite deadly, even if you eat a petal from the plant. The very toxic, I guess it's the root or tuber of the plant, but also is important in medicine. So, what can you share with us about how colchicine is used and what you're studying on it?
Yeah. So colchicine, as you mentioned, is produced in autumn crocus. It's also produced in a plant called flame lily. This is the plant behind me in this picture. It's actually a signed picture from my lab mates from when I was in Beth Sally's lab. I was working on colchicine and using the flame lily as a model for trying to understand how that was made. So it was a sort of nice parting gift when I left the lab. Um, yeah, colchicine is one of these really ancient medicines. You can find descriptions of autumn crocus or plants that people are pretty sure autumn crocus and Eber's papyrus, which is I think like 3,000 something years old. I forget the exact date on that, but many thousands of years old. And this is a medical text essentially saying like, you should take this remedy or this botanical medicine in order to treat this particular condition. In the case of colchicine and the plants that produce it more specifically, they've served as a historical treatment for different types of inflammation. And I think that's how it was described, its use in Eber's papyrus. And colchicine itself is currently FDA approved for the treatment of gouty arthritis. So this is an anti-inflammatory molecule, but as you mentioned, it can be quite toxic. And part of that is that a well-known mechanism of action associated with colchicine is that it's a mitotic poison. So it inhibits the ability of cells to divide. You can imagine that if you have actively growing cells in your body that are quite important, and all of a sudden you dose yourself with colchicine, those cells will not divide anymore. This could be quite toxic. Obviously, this is also the mechanism of action for many chemotherapeutics, and so it's all dosage dependent. So you can safely take colchicine at certain concentrations, but too much can definitely be poisonous.
As Periclesis say, the dose makes the poison, right? But a lot of our important medicines obviously are poisons if taken in the wrong dose. I mean, even caffeine can be a medicine and a poison. So yeah. So what have you learned about colchicine's biosynthesis? Does it have many different enzymes involved in its production, or yeah, what can you share with us?
Yeah, so this ended up being a many-year effort. It was initiated by a graduate student in Beth Sally's lab, Warren Lau, and I picked up the project halfway through, something like that. And he had made a ton of progress, but it's a pretty complicated pathway. So we know in hindsight that it takes about 20 enzymes to make this molecule from two amino acids. So amino acids are the building blocks of proteins. These are present in all organisms. In the case of colchicine, it's derived from phenylalanine and tyrosine, two different amino acids. And so it takes 20 enzymes to convert those all the way through to colchicine itself. And I think this ended up being a very useful case study for the process of pathway discovery. I talked earlier about how it's really important to first figure out where and when the molecule is being made. In this case, it was being made in the rhizome of the flame lily. In the case of autumn crocus, it's made in the bulb, also in the seeds. But then we could correlate that production with the expression of genes in the plant. And then we could use that information to try to prioritize candidate genes that we could then test for function. So again, here, I guess we're just to emphasize what we're looking for are genes that encode for enzymes, these little protein machines that can catalyze chemical reactions. And one of the things that you have to do here, once you're correlating gene expression, production of a molecule, you actually need to pick out the right types of enzymes that can actually catalyze the reactions that are necessary in your pathway. And there's a lot to go into here, but a simple way to think about it is an example of, well, if you want to install an oxygen atom onto your molecule, there are certain families of enzymes that are very good at doing that. And there's certain enzyme families that don't do that at all. And so when we look at this genetic information from the plant, we're trying to tease out what we think are reasonable enzyme families that could catalyze the chemistry that we think is happening. So this requires us to build a chemical hypothesis for how we think the molecule is being made, and then try to assign different enzymes that could catalyze that. And that's how we pick candidate genes out of the many thousands of genes that are present in the flame lily. And then once we have these gene candidates, we actually need to test for their function, right? It's one thing to find a gene and have a prediction of what it does. It's another thing to actually confirm that at a chemical level. And so what we do is leverage this system called agrobacterium-mediated transient expression, which is a mouthful. But basically what we do is we can clone out, isolate a gene from the flame lily, and then move it into this plant pathogen called agrobacterium. And what this pathogen does naturally is infect the plant and actually insert some DNA into plant cells. And people have figured this out a long time ago. You could put whatever DNA you want into that agrobacterium, and then it will inject that into plant cells. And then you'll have the plant actually express your genes of interest. So it's sort of like your way of producing a protein that you want to study. In our case, for our actual production platform, we use a relative of tobacco, Nicotiana benthamiana, and we can pretty easily take genes and move them into this tobacco plant. And so I think one of the pictures I shared with you is a tobacco plant expressing green fluorescent protein. So you could take just a single protein that fluoresces and move it into the plant, and you get this nice green fluorescence. So the plant is producing protein. You can also do this with biosynthetic pathways. So I have another picture that you have this like nice red splotchiness that's on a tobacco leaf. And that's the result of moving all of the genes necessary for producing the pigment that make beets red. So that pigment is a small molecule. And so if we put in all the genes necessary to make that molecule, we can make tobacco plants turn red like beets. And we're not interested in the beet metabolites necessarily, but it's a demonstration of how we actually test for enzymes in the system.
That's incredible. So, for those of the listeners that maybe can appreciate the complexity and the cool science here, but maybe they don't fully understand the relevance to everyday people. And I'm immediately thinking of, medicinal product production. Can you tell us how this kind of knowledge is important to the manufacture of plant-derived small molecules can become like pharmaceutical drugs?
Yeah, definitely. I mean, I think at a basic level, one of our motivations as a lab has always been, really cool, interesting chemistry, but it's also directly connected to the utility of these molecules and rationale for why we should study a specific molecule in the first place. There's hundreds of thousands, if not millions of molecules in the plant kingdom. Why pick one over the other? For many of these plant compounds that are useful as medicines, there's often major limitations in actually being able to source them. And this could be because the plant that produces them is slow growing, and so it's not practical to cultivate them. In many cases, plants are harvested from native ecosystems in order to get the molecule. And so this is damaging to the ecosystem itself and also can lead to endangerment of specific species. This is actually the case for the Cuperzia plants that I mentioned before that make cuperzine. The main way that molecule is sourced is actually just going out into the forest and harvesting these plants that can take decades to grow to maturity. You also have other complex issues like your molecule might be one among thousands in a given plant. And so even if the plant produces it at relatively high levels, you still have to purify it away from that plant, which can be expensive, laborious, and sometimes not practical. And so one of the reasons we would actually want to find these biosynthetic pathways is that if we know all of the genes that encode for the enzymes, people have already figured out ways to take these and move them into something like baker's yeast or E. coli or some other easy to grow micro or other organism that's easy to grow and actually build the molecule from a medicinal plant in some other system. And if we're able to do this successfully, you could alleviate pressures on a plant that's endangered or source a molecule that's otherwise very difficult to source. And you alluded to this earlier. Many of these molecules that we focus on are really structurally complex, and chemists can often synthesize them using traditional organic chemistry. And while this is sometimes possible, it's not always practical. In theory, if we're putting it into yeast, they only really need sugar and some other trace nutrients to grow. We're not necessarily using a ton of toxic reagents or catalysts or things like that. It's a very, think of it as like a green solution for producing these or for accessing these molecules. So that's kind of like the dream scenario. We find a pathway for a useful molecule that's hard to access, and now we have the ability to engineer its production in another system.
That's incredible. So and this, like you said, happens all the time where you can have like a highly valued molecule. I'm thinking in particular of paclitaxel, which is a molecule that women across the world use still today to treat breast cancer. And it grows in a very slow growing yew tree. And so they were able to basically engineer systems, I believe through callus culture is one of the major production schemes now to produce that at scale. So you mentioned yeast. So in this system, you're actually taking the active or the responsible enzymes and moving that into like a yeast-based system to kind of grow in a brew tank. Callus culture, on the other hand, would be taking isolated plant cells with their genes to grow that in kind of a brew tank. Is that about right? Or are there advantages over kind of moving the enzymes into yeast systems over callus culture? Or what can you share about that?
So I think, yeah, you're right about the callus culture being derived directly from the medicinal plant. And in theory, that's a perfectly valid way of doing things. We don't work with plant cell culture in my lab, and I don't really have a lot of firsthand experience working with it. But I know that these are very extremely slow growing. So like plants in general, slow growing. These callus cultures are maybe a little bit faster than a tree, but like not super compared to something like yeast, which if anybody that makes bread out there, like you can observe yeast growing in a matter of minutes to hours when you're doing your fermentation for bread. Um, and so in a sense, it's, yeast are just so much faster growing. I think the actual process of keeping yeast cells alive and happy compared to plant cell culture is much, much easier. Plant cells are prone to being contaminated. If they do get contaminated, these are precious material that took a really long time to grow up in culture versus, yeast gets contaminated. It's a bummer. You can start over the next day. And there's been I think just a ton of engineering effort in yeast to produce different things at scale. So in theory, yeast could be a really excellent alternative in part just because of the systems that are already in place for producing large amounts of molecules.
Do you have any examples off the top of your mind from like yeast products? I'm just not super familiar with it. Are there medicines made through this yeast pathway, or is it kind of more of an evolving science?
I kind of look at it as more of an evolving science and still something. I'm sure that there are good examples. I think particularly for molecules that are maybe slightly less complex. I
Mhm.
think
Yeah.
it's sort of it seems to a certain degree like we're still trying to like reach that sort of like pinnacle where we have this super complicated pathway. We put it into yeast and then it actually becomes an economically viable alternative. And there's all sorts of things that I don't understand that ultimately come to like the business end of things. Like, can you outcompete the current supply chains
Oh,
and things?
yeah.
But I do think that there's really promising possibilities in the near future. So a couple of examples that come to mind, you mentioned paclitaxel or Taxol, the brand name, this chemotherapeutic. Just within the past year or two, there's been a number of papers that have come out essentially completing the biosynthetic pathway or seemingly completing the biosynthetic pathways. It's maybe still a little bit more to understand, but that's a very specific case where if you could put that pathway into yeast, I do think that there would be a lot of opportunity for that to provide this molecule and an alternative source that could, I guess, help with the demand for this chemotherapeutic. Another example is this vaccine adjuvant called QS21 that comes
Mm.
from plain soap bark tree. And this is,
Oh, was this used in the COVID vaccine? I think I remember
that's
something
right.
about that.
It's
Yeah. Okay.
formulations, I think, in the Shingrix vaccine. I think it's being explored in many others. And this is an extract that comes from the bark of the Chilean soapbark tree. So this comes from trees. And there are, I think, tree plantations that supply the molecule currently, but as you can imagine, being very slow growing, if there's any sort of environmental calamity or something like that where these trees are growing, then that's the supply chain very quickly goes away. So within, and maybe it was last year, something like that, year and a half ago, there were a series of publications that found the complete pathway for QS21, the molecules in QS21, and then put that into yeast and demonstrated they can produce this molecule in yeast. And they actually showed that while they couldn't produce a ton of the molecule, the levels they were producing was actually on par with the amount that the trees make, especially considering how much faster yeast is growing. And so I don't know where that's actually at in terms of development, but that actually seems like a really exciting space where there's this molecule that's really complicated from the Chilean soapbark tree that you can actually maybe just produce in yeast. And that might just be the source moving forward. And I think those two cases are extreme in the sense that the molecules are very complex, very long biosynthetic pathways. And so I'm looking to them in terms of if those work and are major success stories, and it almost feels like anything's possible. Cautious with optimism here.
I mean, but I think you're absolutely right. I mean, that would be the ultimate like proof in the pudding because those are such difficult molecules to synthesize or produce through callus culture. I mean, there's been many different academic and industry scientific groups working toward production of some of these key medicines. And I really like what you said too about the realities of, can we grow our way out of this? We have, as the human population expands, we have more and more limited land mass. And I think the other challenge, at least I see on the growing side, is that plants will produce variable levels of some of these molecules depending on all kinds of environmental factors. Like, did they get too much rain? Or was it cloud heat? Or were there certain insects that were or were not there that kind of could influence the whole plant's ability to produce certain molecules? So I think one thing that's very attractive to me would be the level of control you could have in manufacturing of these important medicines with these technologies.
For sure. I think, being able to put something into a vat and know like all of the chemistry of the pH and the temperature and for growing your yeast, I think there's maybe a sense of security in that. But I think in the end, it may end up just being a combination too, where maybe the Chilean soapbark tree, where it's currently being grown and harvested, is still a major source of the molecule. But then you also have this additional source that could be useful in the event that there's a bad year for production or there's some sort of supply chain issue. Um, whether it's environmental or some sort of other global supply chain issue. And maybe just having multiple sources that are viable would just be useful in terms of ensuring that there's enough for all of the need, whether it's for a vaccine or for a chemotherapeutic.
That's amazing. Well, I tell you, like Brian, like I said, I love the work that you do because, in my end, in my lab, we're more about discovery of interesting blueprints to molecules. And I have no idea how the plants make them, right? I find them, we pluck them out, the needle out of the haystack, figure out how it works in a phenotypic system as we try to develop new medicines. But this is such the crucial next phase in how you can achieve then production of some of these hopefully transformational medicines in the future. I know we're almost out of time, so I just want to end with just asking, how do you pick your next projects? What's kind of driving your curiosity now in the lab? Are you going to be continuing to work on huperzine A and colchicine? Do you have your eyes on other classes of molecules besides alkaloids? I mean, there's so much you could do just on alkaloids alone because they're so structurally diverse. But yeah, what's inspiring you lately?
Yeah, there's a couple of directions I tend to think about and I tend to push students to think about when they're picking projects. One always goes back to the chemistry. I mean, I think fundamentally the people in my lab and me included are just very motivated by finding new examples of chemical reactions and enzymes that catalyze those reactions in the plant kingdom. Part of this is, basic discovery endeavor to try to, I don't want to say catalog. I feel like that maybe doesn't come across quite the right way, but really just figure out what chemistry is possible in the plant kingdom. We know a lot about certain types of pathways in plants, but as we explore new pathways and new structures of molecules, it seems like we always find something new in terms of the chemistry, a new enzyme, a new chemical transformation that hasn't been observed before. And I think understanding all this is very important because we can think about engineering pathways for specific molecules that we know exist, but you could also think about taking these enzymes and using them like a toolkit to build any molecule that you want, or within reason, any molecule that you want. And so I think in a sense, cataloging all of this chemistry, exploring different molecules from different classes and different plants is quite important for figuring that out. And it also sort of, satisfying the basic level in terms of new and interesting chemistry. I would say beyond that, I mean, we work on many molecules that are neuroactive or psychoactive. And I think we're increasingly becoming interested about the why for that. Why do plants make so many different psychoactive molecules? Is it really as straightforward as, it's just purely there to hit animal nervous systems and defend themselves, or is there something much more intricate and interesting going on? And I think the fact that there's so many different molecules, different structures that hit the nervous system in different ways, it sort of begs the question for much more subtle interactions that might involve manipulation. And I mentioned before, like the caffeine and the nectar of citrus trees that can be maybe used to convince bees to come back more often than not to pollinate. I think we're starting to think a lot more about why the plants make these molecules and how that might impact organisms that would interact with that plant in a native environment, with the thought that this might lead to some more fundamental insight of what the molecule is actually doing. One case or one example that I'll give that's actually not truly plant related, but I'm actually like really fascinated by is I'm co-mentoring a student in my lab that studies a caterpillar, and this caterpillar secretes essentially a nectar from an organ on its back, and ants will come and eat that nectar. And partly because of the nectar, probably, and some other factors, instead of coming and attacking the caterpillar and eating it, these ants will just take care of the caterpillar, keep it safe from parasitoids. And the student is looking into molecules that are in that nectar to see what are these molecules? Are they potentially manipulating the behavior of these ants? I think there's really countless examples of this in nature. We focus mainly on plants, but just so many cool ways in which small molecules and chemistry is being used for organisms to interact. I think these are the areas that we're really excited about, these sort of like very specific interactions, maybe between a plant and an insect, in this case, and insect and another insect. But I think these are really exciting areas. I think that it will lead to new discoveries that could be relevant to medicine. We always look at the same targets. Um, for drug targets, you're going to find maybe many of the same things. If you look into these interesting new systems, maybe you'll find something different.
No, I completely agree. I think the ways to make paradigm shifts in science is really to think about, are we asking the right questions? How do we get out of this perspective of the box? If the box is, a certain target or system, like to really think about where does the science lead us? And I think this is a really promising area where I'm sure we're going to see in our lifetime some incredible advances in how we discover and manufacture medicines for the public good. So thank you for all the work that you do and for sharing that with us today, Ryan. This was really great.
Yeah, thanks again for having me. It was a super fun conversation. I hope that what my lab is doing is able to contribute to some advance in medicine. I think plants, excellent chemists. I think we've barely tapped into the chemistry in the plant too, and there's a lot to be offered. So hopefully we can contribute to that. And yeah,
100%. Yeah, I'm sure you will. I mean, this is where the future is for these production pathways. So very exciting. All right. You've been listening to Foodie Pharmacology, the science podcast for the food curious. You can tune in on our website at foodiepharmacology.com or head over to our YouTube channel at Teach Ethnobotany to watch the video version. We've been talking a lot about alkaloids on this episode. This is just your gentle reminder. I also need some alkaloids. You can help us out by buying me a coffee. You can do that at buymeacoffee.com/foodiepharma. You can find the link also on our website and on the video QR code on YouTube. Thanks so much for listening. Stay healthy out there, and we'll see you next time.